I'm a clinician. So a couple of times a year, I'm the cardiologist for the hospital. And so I spend some time at the bedside. And so my lab, rather than the traditional bench to bedside approach, I have in general taken the opposite approach. And that is to find some observations at the bedside that we know to be true in humans, but for which we really don't understand the molecular mechanism, and take those back to the bench and really try to figure out what's going on from a molecular standpoint that will explain the observations, so that the next time that I see patients and I'm standing at the bedside, I'll have a better understanding of what's actually going on in that patient, with the ultimate goal, of course, of providing better patient care. So let's start with some observations that we know to be true in terms of sex differences in cardiovascular disease. So the one that everyone knows is that in premenopausal women, there is protection from risk of heart attack compared to men. So this is new heart attack or fatal coronary heart disease per 1,000. And you can see that in premenopausal women, they're relatively protected. Then menopause happens, and it takes about a decade or two, and women ultimately catch up to men in terms of their risk of heart attack and cardiovascular death. And actually, with advancing age, women exceed men in the risk of heart disease and cardiovascular death. And the mechanism for those is not completely understood. So what is another observation that we know? So obesity is a worldwide epidemic and is rapidly increasing and disproportionately affects women. So 38% of men worldwide are overweight or obese, 40% of women, and in the US, it's even higher than that. And why is that important? Because obesity, particularly when it's associated with metabolic dysfunction or diabetes, mitigates the cardiovascular benefits of female sex. So here's a couple of observations that lead to that conclusion. So although premenopausal women have decreased risk of heart attack compared to men, the incidence of heart attack in women aged 35 to 54 has been increasing, while the risk in age-matched men is declining, and actually in every other age group in both sexes is declining, but only in women ages 35 to 54 is the risk rising. And that rising risk is associated with a simultaneous increase in Framingham risk score, with women having an increased incidence of obesity and diabetes. So the thought is that that's driving this increasing risk in premenopausal women. And we all know that obesity impacts both males and females in terms of cardiovascular risk, but the impact of obesity on cardiovascular risk is greater in females, particularly when associated with metabolic dysfunction. And then the final point on here is that obesity is associated with increased levels of the hormone in plasma, aldosterone, which is a hormone that we know regulates blood pressure, that I've been studying for almost 2 decades now. And the rise in aldosterone in obesity is even greater in females. And so that's actually what got me interested in some of these questions. So a little bit of background then on the hormone aldosterone and its receptor here in the kidney, the mineralocorticoid receptor, or the MR. So it's been known for half a century that this pathway is critical to regulation of blood pressure. And just to remind everyone, what happens is when blood pressure is low, that's sensed by the kidney, renin is released, a proteolytic cascade is initiated, resulting in angiotensin II production, which acts on the adrenal gland to promote the release of the hormone aldosterone. The hormone aldosterone is a steroid hormone like estrogen and testosterone, so in the same family. And aldosterone is known to act in the kidney by binding to the mineralocorticoid receptor to promote salt and water retention. And so, of course, when salt and water is retained, blood pressure rises, addressing the lower blood pressure. But it's also known that activation of this pathway is associated with increased risk of cardiovascular disease. And that has had for a long time been associated with increased blood pressure, because we know that increased blood pressure is a risk factor for heart disease. But what my labs have worked on for a long time is direct effects of aldosterone and the mineralocorticoid receptor in the blood vessel, and how they might directly contribute to cardiovascular disease. And what were the observations in the clinic that made us actually think to even look at that? So here's a paper from a French group in 2005 where they took patients with primary hyperaldosteronism, so people with a tumor that makes too much aldosterone. Not surprisingly, those folks have hypertension. And if you match them to 465 patients with the same degree of hypertension but without high aldosterone, turns out having high aldosterone results in a 4-fold, so this is not 40%, this is 400% increased risk of stroke, and a 6.5-fold increased risk of non-fatal heart attack in patients with high aldosterone associated with high blood pressure rather than normal. Now, you might argue, well, these people, primary hyperaldosteronism, it's not that common, although not that rare. and their aldosterone levels are really high. But what if they're just in the high end of normal? So it turns out that is also associated with a profound increased risk of cardiovascular events. So this was a study, much more recent, with patients coming to the cath lab. So we take care of patients as cardiologists who have blockages in their heart arteries. So these people were coming in for elective angioplasties of blockages in their heart arteries. So they have underlying cardiovascular disease. And all they did is when they came to the cath lab to have their procedure done, they measured their serum aldosterone level. And then they followed them for 16 months. And if the serum aldosterone level was above the median, so still in the normal range, they had a 3.5-fold increased risk of dying of a heart attack or cardiovascular death in 16 months. And if they survived, they still had a 2.4-fold increased risk of having either a heart attack or stroke in the next 16 months. So having increased aldosterone is associated with a very high risk of having a heart attack or stroke. And obesity is associated with increased aldosterone levels. So we wanted to take all of these observations, the sex differences, as well as the aldosterone effects, back to the bench and understand what's the mechanism so that we can have a better understanding of what's happening in our patients. So when you go back to the bench, the first thing is what's already in the literature, so what's known. So this slide is the simplest one I could find about what's known about how this pathway works. So aldosterone is a steroid hormone. So like all steroid hormones, it's lipophilic and it diffuses through the membrane. It binds to an intracellular receptor, in this case, the mineralocorticoid receptor. And it can act in genomic, classical ways or non-genomic ways. And the genomic mechanism is that it goes to the nucleus, binds to DNA sequences, and increases transcription of target genes. And in the kidney, those are genes that retain salt and release potassium, thereby regulating salt and water homeostasis. And then there's also non-genomic pathways that are mediated by cytoplasmic mineralocorticoid receptors, and then also potentially alternate receptors that lead to rapid signaling pathways that I'll talk about at the end. And just to remind everyone, there's 2 drugs that are already approved to block this pathway. So spironolactone and eplerenone prevent activation of the mineralocorticoid receptor, and they're used to treat high blood pressure and heart failure. And there's a whole new class actually of non-steroidal mineralocorticoid receptor antagonists that are in late phase clinical trials. So there will be more. So if we want to understand how they could lead to heart attack or stroke, what is the mechanism or what do we know about how heart attack or stroke occurs? So there's a huge literature and I will try to summarize it in 1 minute. So when the lining of the vasculature becomes damaged, so these are the endothelial cells that line the vasculature, they become dysfunctional. And the dysfunctional endothelium expresses adhesion molecules on their surface, which allows white blood cells to adhere and migrate into the subendothelial space. So these endothelial cells can become damaged by diabetes or hypertension or smoking or any of the other cardiovascular risk factors, or cancer drugs, which is the other half of my lab studies. So that they become pro-inflammatory. Inflammatory cells migrate into the subendothelial space. They take up cholesterol and they form atherosclerotic plaques. On top of the plaque are smooth muscle cells and a fibrous cap. And these inflammatory cells secrete or release factors that recruit other cells like T cells. And together, those all release matrix metalloproteases. And when those proteases degrade the fibrous cap, the cap can rupture. And if it ruptures, a thrombus forms. And now a blockage like this that's blocking only half of the vessel, it ruptures and a thrombus forms. And now it's suddenly blocking the entire vessel. And if that happens in a coronary artery, it causes a heart attack. And if it happens in a cerebral vessel, it causes an ischemic stroke. So we were interested if patients with high aldosterone are more likely to have heart attacks and strokes, whether or not aldosterone promotes atherosclerosis. So this is decades ago now. So Adam was in my lab like early 2000s. He did the first study that we ever did in an atherosclerosis model. So he took the classic apolipoprotein E knockout mouse, and it's a mouse that develops high cholesterol and atherosclerotic plaques that look somewhat like those in humans. And they develop plaques spontaneously, but if you put them on a high-fat diet, they develop them faster. And so he put them on a high-fat diet. He calls it the Cheetos experiment because the high-fat diet are these little nuggets that are kind of yellowish and they look like Cheetos. So he puts them on the Cheetos and then he randomizes them to infusion of placebo or aldosterone. And after 4 to 8 weeks, he looks at their blood vessels. So if you take out the whole aorta and stain it with Oireto for lipids, you can quantify the amount of plaque globally, and that's increased by aldosterone. And of note, the dose of aldosterone we used did not change blood pressure, so it was a pretty low dose. And then if you section through the aortic root, which is sort of the traditional way we look at atherosclerosis, you can see here's the aortic valve with the 3 leaflets. And this right here is an atherosclerotic plaque. And with oil red O, you can see the lipids in red, and it's not hard to see that the aldosterone-treated plaques are bigger and they have more lipids. But more importantly, Adam took the aortic arch of these animals. So here's where the aortic root would be, and then here's the arch, and then these are the three great vessels. And he digested that aortic arch to a single cell suspension and did flow cytometry to quantify the number of inflammatory cells. And what you could see is that the animals treated with aldosterone have substantially almost twice as many inflammatory cells in their aortic arch. And you can parse those out into T cells and myeloid cells, and all of them are increased. And the converse is also true. So this is a paper by a different group, but they showed that if you take ApoE knockout mice and treat them with eplerenone, so a mineralocorticoid receptor antagonist, the size of the plaques gets smaller. And Elizabeth Moss, an MD PhD student in my lab, more recently did the same experiment and looked at inflammatory cells, and you can see that a mineralocorticoid receptor antagonist, spironolactone, results in a 50% decrease in inflammatory cells in the aortic arch. And this is important. This is important because plaques that are inflamed are more likely to rupture. So here's a plaque from someone who died of a heart attack, and here's their plaque core, and here's the fibrous cap, and here's where it ruptured, and a clot formed in their coronary artery, and they died from that, and that's why we have the histologic specimen. And from looking at pathology specimens from people who died of heart attacks, it turns out that more inflamed plaques are more likely to rupture. So this got us interested in whether or not mineralocorticoid receptors directly in the vasculature contribute to cardiovascular disease through enhancing inflammation. So how might they do that? So first we had to look at the cells of the vasculature and see whether or not the mineralocorticoid receptor was expressed there. At the time that we started these studies, so this is published in 2005, it was well known that the receptors are expressed in the kidney, but where else they were expressed and what they do was really quite not well known. So to remind everyone, the blood vessel is made up of several layers. So the innermost layer is the endothelium, and it's a single layer of cells that line the vasculature and is in contact with circulating blood. The endothelium is surrounded by several layers of smooth muscle cells that can constrict and relax to change the diameter of the vessel. And that's surrounded on the outside by the adventitia, which is made up of fibroblasts and extracellular matrix. So the first thing we did is just look in human tissues and cells and ask whether the mineralocorticoid receptors expressed. So this is human aorta and coronary artery tissue, so the whole thing ground up. And you can see there's the mineralocorticoid receptor expression. And when we cultured smooth muscle cells from them, there's plenty of mineralocorticoid receptor in smooth muscle. And then when we looked at endothelial cells from coronary or aorta, as well as from venous sources, the mineralocorticoid receptor was expressed. And we've actually done most of our work in understanding what the mineralocorticoid receptor in smooth muscle cells do, but I'm going to focus the talk today exclusively on the endothelial cells because that's where we have a lot of more information about sex differences. So Kaitley Barrett-Mueller, who was an MD PhD student in my lab, was interested in understanding what the mineralocorticoid receptor in the endothelial cell does. So she created a mouse model where the mineralocorticoid receptor was deleted specifically from endothelial cells. And she did this using the VE cadherin Cree in a floxed mineralocorticoid receptor mouse. And here she took cells, so mouse lung endothelial cells and mouse heart endothelial cells from those mice and showed a profound reduction in mineralocorticoid receptor expression. And she looked at leukocytes in the spleen and showed that in our model, there was no deletion of the mineralocorticoid receptor from white blood cells. So that's important because if we want to understand the role of the endothelial cell in inflammation, we wanted to make sure that the white blood cells had the mineralocorticoid receptor intact. And after she made that mouse, Elizabeth Moss came to the lab and she was interested in the role in atherosclerosis. So this new model had just been published at the time where you could take an adenovirus expressing a constitutively active mutant of the human PCSK9 gene. And if you injected that into a mouse, it would go to the liver and express PCSK9, which degrades the LDL receptor. And so you would basically make an LDL receptor knockout mouse with a single injection. So Elizabeth took these endothelial specific MR knockout mice, gave them the PCSK9 and put them on a high-fat diet and did the Cheetos experiment to see whether or not the mineralocorticoid receptor in endothelial cells contributes to atherosclerosis. So she took males and females, and so this is where we discovered sex differences. And I'll just say, anytime we've ever looked at males versus females to see if anything is different, it's always different. So the answer is if you look, you will find a sex difference. So this was one of the first experiments actually that we did in males and females side by side. So the mineralocorticoid receptor is intact or knocked out in males, and it's intact or knocked out in females. We make them hypercholesterolemic. We put them on high-fat diet for 12 weeks, and we look at their aortic roots to see what changed in the size of the plaque. And what you can see here is that, first of all, the genotype had no impact on the size of the plaque. So they're not bigger or smaller in the presence or absence of the mineralocorticoid receptor. And what you can see, which is even more obvious, is that females have bigger plaques than males. And that was no surprise. So in all of the mouse models, and actually not too many, many of the other small animal models of atherosclerosis, females have bigger plaques than males. And we recently wrote a review article, actually, where we reviewed every model. And if you want to see it in a table, it's in the review article. And this has been known for a long time, and based on very early studies in both the ApoE and LDL receptor knockout mouse showing that females have bigger plaques, has led to the rationale that we shouldn't use these models to study females, because we know females have fewer heart attacks than males, so it doesn't make sense that their plaques are bigger, so it's a bad model for female disease. But I'm going to continue and suggest to you that perhaps plaque size is not the most important variable, and that perhaps this model is useful to study disease in females. But what we can conclude here is that females have larger plaques and that the presence of mineralocorticoid receptor in the endothelium has no impact on plaque size. But Elizabeth went further, and she took the aortic arches and digested them and did flow cytometry. And that's where things became interesting. And so I'm going to start with the males. So the blue are the males. And what we saw was that if we look at total leukocytes, so CD45 positive cells, there's about half as many in a male mouse that lacks mineralocorticoid receptor in the endothelial cells. And if we parse that out into monocytes and macrophages, we see the same thing. So half as many monocytes and macrophages when you delete the mineralocorticoid receptor from the endothelium. But when Elizabeth looked at the females is where it got interesting. So just in the MR intact females, right? So this is intact males and intact females. There was half as many inflammatory cells in females, atherosclerotic plaques, compared to males. And those females look quite similar to males that lack the mineralocorticoid receptor in their endothelium. And remember, I just showed you that the female plaques are twice as big, but half as inflamed. So if inflammation is what leads to plaque rupture, there's 75% less inflammatory cells relative to plaque size in females, at least in this model. And the other thing to note is that when you delete the mineralocorticoid receptor from endothelial cells in females, it has no further impact. So what could be the mechanism for that? So we had previously published a paper in 2008 showing that human coronary artery endothelial cells express the mineralocorticoid receptor. It can be activated by aldosterone. And when we looked for target genes, we just sort of screened, we found one of the target genes to be ICAM, or intracellular adhesion molecule 1. And ICAM is an inflammatory marker that's expressed on the surface of endothelial cells and allows leukocytes, or white blood cells, to interact with the endothelium and move into the vasculature. So Elizabeth was interested in whether or not mineralocorticoid receptor in endothelial cells plays a role in leukocyte trafficking into the vasculature. And she looked at that by intravital microscopy. And of course, there's a lot known in the literature about what is involved in leukocyte trafficking. So it's been actually quite well worked out. So the normal healthy endothelial cell is anti-inflammatory and doesn't express adhesion molecules. And the leukocytes circulating just fly by very quickly and don't interact with the endothelium. But when there is an infection or an injury, or in this case, the cardiovascular risk factor, the endothelial cells express selectins, so P-selectin and E-selectin, and then CAM, so ICAM and VCAM. And the selectins play a role in leukocyte rolling. So ligands on the surface of the white blood cells bind to these selectins, and it allows them to slowly roll across the surface. At first, they roll quickly, then they roll slowly. As they roll slow enough, then their integrins are able to interact with the cell adhesion molecules. They firmly adhere, migrate into the subendothelial space, and the process of vascular inflammation begins. And you can study all of this by intravital microscopy. So if you look at this video, this is one that Elizabeth did. So she injected a fluorescent label to label all the circulating cells, and then she imaged. These are small vessels in the mesentery. And so you can see that there's some cells going by so fast you can't even see them, and those are not interacting with the endothelium. But you can clearly see that there are white blood cells that are slowly rolling, quickly rolling, and then the one that's in a dashed circle is a firmly adherent cell, so it sits in the same place for the entire video. So Elizabeth did this in our 4 groups of mice, and then she quantified in those images the number of fast rolling cells, the number of slow rolling cells, and then the number of firmly adherent cells. And while there was no significant difference in fast rolling cells, the number of slow rolling cells had a pattern that was the same as what we saw for vascular inflammation. And that is, in males, There's about half as many slow rolling cells. The females, where the mineralocorticoid receptor is intact, have fewer slow rolling cells, and the deletion of the receptor has no further effect. And this similar pattern was seen in terms of adherent cells. So MR intact males have more adherent cells than knockout mice. Females look like the knockout mice, and deletion in the females have no effect. So if slow rolling is mediated by E-selectin, we looked at the expression of E-selectin in those vessels in the various groups. And what you can see by Western blot is that E-selectin protein expression is decreased in males lacking the mineralocorticoid receptor in their endothelium. It's decreased at baseline in MR intact females, and it doesn't change further when you delete the receptor. And the same is true for ICAM. So male mice lacking endothelial mineralocorticoid receptor have less ICAM, and so do the females, regardless of genotype. So putting it all together, how do we explain the plaque inflammation information in the mice? So what we think is that in the human coronary endothelium, the mineralocorticoid receptor is driving ICAM expression and E-selectin expression, and that's causing white blood cells to adhere and making plaques more inflamed. We think that the estrogen receptor, with its hormone estrogen, binds to the mineralocorticoid receptor in the nucleus and blocks this. And what is that based on? So we actually published an entire paper. I'll show one piece of data where we actually looked at how the estrogen receptor and mineralocorticoid receptor work together on DNA to regulate ICAM expression. If you delete the mineralocorticoid receptor from endothelium in males, there's a decrease in expression of these molecules and less inflammation in the plaque. Whoops. But if you delete it in the females, since it's already been inhibited, there's no further impact. And here's one piece of data from this paper in endocrinology in 2014, where we showed in endothelial cells from humans, aldosterone increases ICAM expression, and that's blocked by estrogen. And ICAM increases leukocyte adhesion. This is in a static leukocyte adhesion assay, and that's blocked by estrogen. So how does this help us explain what's happening at the bedside to patients? So we think that vascular or endothelial mineralocorticoid receptors drive inflammatory adhesion molecule expression in the vasculature. It makes plaques more inflamed. We know more inflamed plaques are more likely to rupture, and plaque rupture causes heart attack, stroke, and death. We think this may explain why patients with primary hyperaldosteronism have more heart attacks and strokes compared to those with normal aldosterone levels. We also know that premenopausal women are protected from heart disease relative to age-matched men. And premenopausal women have estrogen, and estrogen, through the estrogen receptor, blocks mineralocorticoid receptor regulation of these genes, making female plaques less inflamed and less likely to rupture. But when females become old, they lose estrogen. And so their endothelial mineralocorticoid receptors become de-repressed and now can start expressing adhesion molecules. Not surprisingly, it takes some time. By the time you start expressing adhesion molecules, you recruit the leukocytes, the plaques become more inflamed. By the time they rupture, it's not surprising actually that it's a decade or two later, because the males have been developing these plaques over many decades. So what about obesity? Why is that changed by obesity? So we know obesity enhances activation of the mineralocorticoid receptor, particularly in females, and it does so by increasing plasma aldosterone levels. And other groups have shown that this may be mediated by release of factors from adipocytes that go to the adrenal gland and promote the release of aldosterone. It's also been shown, again, by other groups, that blocking the mineralocorticoid receptor with spironolactone protects against vascular dysfunction in female obese mice, but not in males. So we wanted to examine this further, and we examined both the genomic and non-genomic effects. And in terms of the genomic, the regulation of ICAM by the mineralocorticoid receptor is a genomic effect, and these are primary human aortic endothelial cells from human patients. So we have a bank of those. And so we compared males to females who were young, and what we found is that ICAM1 expression on human endothelial cells is lower in females. But if we take young females that are obese, so a BMI greater than 30 is what we used as our cutoff, it turns out that that is reversed. So ICAM expression is activated relative to non-obese females and similar to males. So consistent with the idea that obesity reverses the beneficial effects of female sex. But what about non-genomic effects, and what about the microcirculation? So it turns out that females are much more likely to have microvascular disease rather than large vessel disease in the coronary arteries. And this is a study from Naomi Hamburg, who's at Beth Israel, and she looked at a large data set of human patients and looked at micro and macrovascular function. And what she found is that female sex was associated with an increased odds ratio of abnormal microvascular function as measured by peripheral artery tonometry. And the other factors that were associated with an increased odds ratio included body mass index, so obesity, as well as diabetes, so metabolic dysfunction. So when Anna Davel came to my lab from Brazil as a postdoc, she was interested in understanding the mechanism for this. And so she took, again, our mice lacking the mineralocorticoid receptor in the endothelium and compared males and females with the receptor intact and deleted. And she had 3 different groups: normal diet, high-fat diet, which led to diet-induced obesity with high glucose. And then she also did a study in hyperlipidemic animals, which I'm not going to focus on today. So we're just going to compare obese and non-obese animals, although some of that data is still in the figures. So first, just to explain how these studies were done. So after the mice had been on high-fat diet for 12 to 20 weeks, Anna took out their small mesenteric microvessels and hung them on a wire myograph in little rings, and then constricted the vessels with phenylephrine, and then added increasing concentrations of a vasodilator, in this case acetylcholine, and looked at how much they could dilate. And acetylcholine-mediated dilation is a measure of endothelial function. So how good are their endothelial cells functioning in the microvessels? So when she did this in control male animals, they dilate almost completely, like about 90%. When they were obese, that's the squares, actually it was unchanged, but with hyperlipidemia, she saw some impairment. When you compare that to females, again, a normal healthy female on a controlled diet dilates about 90% or more. When they were obese, though, you can see that there's an impairment in endothelial function, specifically in the females. When she did the same thing in mice lacking mineralocorticoid receptor in the endothelium, there was no impact in the males. So here's their normal dilation, about 90%. When they're obese, they still dilate 90%. In the females, what you see is that this impairment with obesity is restored in the knockout mice. So she set about to try to understand the molecular mechanism for that. So now it gets a little bit complicated. So in resistance vessels, endothelial-dependent vasodilation is mediated by a variety of factors. So an agonist like acetylcholine activates the endothelial cell. There's a variety of signaling cascades, and the endothelial cell can then release a series of vasodilators that cause the smooth muscle cells to relax and dilate. One of the most important is nitric oxide. A second one is endothelial-derived hyperpolarizing factor, which is actually potassium ions that are released. And then a third actually is prostaglandins and prostacyclins, although in mesenteric resistance vessels, this is predominantly mediated by nitric oxide and EDHF. And you can determine the component of nitric oxide and EDHF by giving an inhibitor. So if you look at dilation and then you add a nitric oxide inhibitor, the component that goes away is the nitric oxide component. If you then add an inhibitor of these potassium channels, then the component that is EDHF goes away. So just to walk you through that, here's just a control. So on normal diet, all their receptors are intact, male mouse. And you could see that their endothelial-dependent relaxation is about 90%. If you give a nitric oxide synthase inhibitor, you lose a little over half. So the area between these two curves is the nitric oxide component. And then if you give an EDH inhibitor, you lose most of the rest, and that's the EDH component. And if you do the same in females, what you can see, and it's a little bit hard to see, is that I think you can maybe see that the EDH box is bigger, a little bit bigger than the males, but overall they're about half and half. So now we did that in males with obesity. Sorry, it doesn't say obesity up here. There it is. So these are obese males. And so one thing that was very interesting is that I showed you before that when males became obese, their dilation was normal. So here is their dilation. But actually, when you give a nitric oxide synthase inhibitor, you see that the nitric oxide component is totally gone, right? So here there's a nitric oxide component, and here there. So these obese males do have endothelial dysfunction. They have a profound impairment in their nitric oxide production, but it's overcome by increasing their EDH, and so they can still dilate. But this is still non-beneficial because there are other benefits to nitric oxide. And when you knock out the mineralocorticoid receptor, this was unchanged. So still a decrease in nitric oxide that's restored by EDH. So the females were quite different. So the females, when they became obese, they had impaired endothelial function, and it was due to a near complete loss of EDH. So this is gone. And the nitric oxide component stays the same. But if you delete the mineralocorticoid receptor from their endothelial cells, they're able to recover some of their vasodilation, but they do so by increasing nitric oxide. So the first take home here is that the males and females respond totally differently to obesity. And in all cases, I think the reason why our microvessels have multiple ways to mediate dilation is to be able to overcome deficiencies. And so when the males become deficient in nitric oxide, they overcome that with EDH. When the females become deficient in EDH, if their mineralocorticoid receptors are not activated, they can overcome that by upregulating nitric oxide. The issue is in obesity, their mineralocorticoid receptors are activated. And that is, I think, one of the reasons why obesity impairs endothelial function so much in females. And you can quantify all of this. So if you actually do the areas under the curve, you can see again that males that become obese have a profound loss in nitric oxide, but overall their dilation is normal. And females have a profound loss of EDH that can be restored by deletion of the mineralocorticoid receptor through increased nitric oxide. And you could actually measure nitric oxide released from the vessels. And indeed, obese males release less nitric oxide than controls independent of genotype. And females do not have a decrease in their nitric oxide release, but if they lack the mineralocorticoid receptor, they're better at being able to respond. So what could be the mechanism for this? So, and this is when we get into non-genomic effects of the hormones. So Richard Karras, who used to be at the Molecular Cardiology Research Institute at Tufts, and Mike Mendelson had done a lot of work on estrogen receptor signaling in the vasculature. And they and others together had shown that estrogen receptor, in addition to its genomic effects on regulating gene expression, also is able to act in a complex of the membrane to activate endothelial nitric oxide synthase by enhancing phosphorylation at this 1177 site, which is an activating site of eNOS. And they further went on to show sort of more about the proteins involved in that. So there's a scaffolding protein called striatin that sits in the caveoli bound to caveolin. And it turns out estrogen receptor can bind to striatin in the membrane. When activated by estrogen, the hormone, the receptor activates a kinase cascade that ultimately phosphorylates AKT, and then AKT phosphorylates eNOS at S1177 and makes more nitric oxide. So we know that estrogen can enhance nitric oxide production. So more recently, Gordon Williams' group, who published this paper, showed that the mineralocorticoid receptor can also bind to striatin in the caveoli, although what it does there was not totally clear at the time. So we were interested in whether or not the mineralocorticoid and the estrogen receptor are interacting with each other in these striatin complexes to regulate nitric oxide production, and particularly in obesity. So to test this, we looked in endothelial cells. So these are human endothelial cells that express estrogen receptor alpha. And we did immunoprecipitation to pull down the striatin protein, the one that sits here in this complex. So pull down the striatin and see whether or not estrogen receptor or mineralocorticoid receptor comes with it. And how does that change with different hormones? So starting with the mineralocorticoid receptor, what you can see is that when you treat with aldosterone, and remember obesity is a setting of increased aldosterone, you pull down more mineralocorticoid receptor with striatin. And with the estrogen receptor, and this had been shown by others, when you add estrogen to endothelial cells, you also pull down more estrogen receptor with striatin. But the important thing is that when both hormones are present, the estrogen receptor basically gets kicked off of striatin and the mineralocorticoid receptor prevails. And we actually, in this paper that was published very recently by Lauren Beware, who's a postdoc in my lab, we went on to show that actually the mineralocorticoid receptor and estrogen receptor compete for binding to striatin in the same region. And so we think that there's a competition there that impacts estrogen's ability to activate nitric oxide in endothelial cells. And we went on to show that. So here we took endothelial cells and we treated them with one of these hormones for a short time. So this is rapid non-genomic signaling, so like 15 to 20 minutes. And we showed that if you treat an endothelial cell with aldosterone, we actually saw no impact on phosphorylation of eNOS. And this is at the 1177 activation site. But if we add estrogen, it has been shown by many groups that there's an increase in eNOS phosphorylation. But what's new here is that this increase in eNOS phosphorylation is blocked by aldosterone or attenuated by aldosterone. And this is dependent on the mineralocorticoid receptor, because if we knock down the mineralocorticoid receptor with an siRNA, and here we show that it was knocked down, then the effect of aldosterone to mitigate estrogen's phosphorylation of eNOS is lost. So then Lauren tried to take that back in vivo and say, does that affect vascular function? And so she made a new mouse where the estrogen receptor alpha was deleted only from endothelial cells on a background where the mineralocorticoid receptor was also deleted in the endothelium. And I already showed you, so this is the nitric oxide component of acetylcholine-mediated dilation in obese female mice. And I already showed you that obese female mice have normal nitric oxide production, but it is enhanced when you delete the mineralocorticoid receptor from the endothelium. But if they don't have the estrogen receptor alpha in their endothelium, then deleting the mineralocorticoid receptor is no longer beneficial and does not increase their nitric oxide. So you need the estrogen receptor to increase nitric oxide in those cells. So putting it into a cartoon fashion, obese female mice with their mineralocorticoid receptors intact, they are exposed to excessive aldosterone because obesity in females is a situation of excessive aldosterone. We showed in the paper actually that the mineralocorticoid receptor also is a suppressor of estrogen receptor expression. So that means that obese females have less estrogen receptor expressed in their endothelial cells in their vasculature. But we also showed that these compete for binding to striatin and for activation of eNOS. And so there's less estrogen bound to striatin, less activation of eNOS, less nitric oxide, impaired vasodilation. If you delete the mineralocorticoid receptor, first we showed actually there's more estrogen receptors in general. More of them are bound to striatin, and they're able to enhance nitric oxide to overcome the impairment in endothelial function induced by obesity. So they have restored vasodilation with an increased nitric oxide component. So let's put the whole story together, right? So what I said at the beginning is that we take observations from the bedside about what we know about patients. We go into the lab and we really delve into the molecular biology. We use animal models, we use human cell culture, and we come up with a mechanism. And then we see whether or not that mechanism can explain what we see in humans. So we know that mineralocorticoid receptors in the endothelium activate expression of cell adhesion molecules and make atherosclerotic plaques more inflamed. More inflamed plaques are more likely to rupture, causing heart attack, stroke, and death. And this is, can explain the association between aldosterone and increased heart attack and stroke. And we actually also know, if you look at clinical trials of mineralocorticoid receptor antagonists, while it's often not the primary endpoint, we see fewer heart attacks and strokes in patients who are put on mineralocorticoid receptor antagonists. So it also suggests that blocking this will prevent heart attack and stroke. In young females, we think that estrogen is inhibiting the mineralocorticoid receptor from upregulating these genes. It makes female plaques less inflamed and less likely to rupture, thereby explaining their increased risk of heart attack and stroke. What happens if a young female is obese? So we know that estrogen is acting in the endothelium to help produce nitric oxide, and that's one of the benefits of estrogen on the vasculature. When a young female becomes obese and their mineralocorticoid receptors are activated by excessive aldosterone, now the ability of estrogen to be beneficial is blocked, both in terms of their adhesion molecules and inflammation, and in terms of production of nitric oxide. And so these obese females actually lose the benefits of estrogen protection, and they have increased risk of heart attack and stroke. And this may be driving the rising risk of heart attack and stroke in females in the perimenopausal or premenopausal age range, particularly those that have increased rate of obesity, metabolic dysfunction, and diabetes. So I tried to thank all the folks involved along the way. So I mentioned Adam and Kate Lee and Anna and Elizabeth, and current folks in my lab working on this are Lauren and Ching. And I have lots of collaborators around the country and around the world, which is part of what's so fun about what I do. And of course, my funding, without which I couldn't do any of this. So thank you all for your attention, and again, for the kind invitation, and I'd really be happy to take questions.
Thank you so much. And yes, for anyone that has a question, if you want to put your name in the chat, we will go down the list. So yeah, really interesting talk. I think a lot in science, we operate in silos where we're only like, what does estrogen do? And what does, aldosterone do? But in the body, that's not reality. So it's really good to see these crosstalk that happens. So I guess I wanted to ask, while people are thinking about their questions, if you looked at there is some people that report that there's interaction between the MR and G protein coupled estrogen receptor. If you have looked at whether, in your knockout model that receptor was affected, or if you saw any other changes.
Yeah, so that becomes even more complicated. So the short answer is we have not looked at G protein coupled estrogen receptor at all, or GPR30. And of course, and in fact, I also showed you only estrogen receptor alpha is what we deleted, but of course there's also estrogen receptor beta. And I can say that when we looked at the genomic regulation of MR on the ICAM gene, we actually looked at both ER alpha and ER beta, and they both seem to be able to go to the nucleus and affect the genomic actions. But GPR30 acts in the cytoplasm and mediates the non-genomic effects of estrogen receptor. And the short answer is we haven't looked, but it's even more complicated because there is a literature about aldosterone activating GPR30 in addition to estrogen. So you can imagine, therefore, in the cytoplasm for these non-genomic effects, you have MR and ER competing to bind to striatin. And then you have GPR30 or GPER that could potentially be activated by either of the receptors. I think the literature for estrogen binding to GPER is much stronger than the literature for aldosterone actually binding to GPER. And my own opinion is that I think that aldosterone may not actually bind to the receptor, but that it does act downstream of the mineralocorticoid receptor, in which case there certainly will be an interaction between estrogen actions and aldosterone actions via GPER. And we haven't looked at that. So as you point out, people tend to look at their favorite receptor and not at the interactions. And here I show you the interactions between two, but you could always add the third and the fourth and the fifth. There's many, many more interactions taking place. So that's a great question.
So I think Dr. Mave Jarvis has a question. Do you want to come on, Frank, or you want me to read it?
Is it helpful for me to stop sharing my screen?
Sure, yeah, go ahead and stop sharing.
Very nice talk. I was wondering, so you showed clearly that the MR, is in the nucleus. It can signal the membrane, the nose, etc. But can it be in the mitochondria and signal in the mitochondria too? Is it localized in the mitochondria?
Yeah, so to my knowledge, I don't know anything literature about MR localizing to the mitochondria. The one thing I do know is that the specificity of the mineralocorticoid receptor for binding or being activated by aldosterone, as opposed to glucocorticoids, which can also bind to the mineralocorticoid receptor, is mediated by the enzyme 11-beta-hydroxysteroid dehydrogenase 2, which inactivates cortisol. And that enzyme sits in the inner mitochondrial membrane and inactivates cortisol. So that is the one mechanism by which what is happening in the mitochondria affects the activation of the mineralocorticoid receptor, but I'm not aware of the MR acting in the mitochondria.
Thank you.
Hey, Dr. Agola.
Thanks, Dr. Jaffe, for the presentation. And I think your work fits well with what Lisa Cassis had shown and current rules. So going back to sex differences. So you've shown really the hormonal component. However, there's the chromosomal component that contributes to sex differences. Have you or in any way thought about fitting the MR component to sex chromosome component in atherosclerosis or vascular inflammation? Because what Lisa Cassis is showing is excess complement. In the absence of sex hormones, you have these exacerbated atherosclerosis plaques.
Yeah, so again, the short answer to the question is we have not looked at the chromosomal component at all. But that being said, I had mentioned that whenever you look for a sex difference, whatever is the outcome you're looking for, we always find one. And in general, I think that those differences cannot be exclusively explained by hormones, right? So we, like others, have done the experiments where you remove the ovaries and then you give back estrogen and you see how much of the difference you can replace. And it's usually insufficient. So clearly there are other things going on besides just the hormonal component. It's just it's more complicated or difficult to actually tease that out. right? And the chromosomal component, there's many genes involved that are on the chromosome. And I'll add one more layer, which I talked to a few folks about this morning. So what could be mediating sex differences? So one thing is hormonal differences, one is chromosomal differences. And then the other thing I'll add is just that each of these changes, whether it comes from a difference in the chromosome, what's on the X or Y chromosome, or the hormone, initiates a cascade of events that moves forward downstream, right? So the other thing is, even if you remove the ovaries from either an animal or from a woman who becomes postmenopausal, you didn't remove the fact that an entire series of events took place over months, weeks, decades, that estrogen was there. And that led to downstream effects that still makes that individual different than they would have been, even if they no longer have estrogen. And so how to integrate all of what changed over time, secondary to those things that changed throughout the premenopausal period, is something that I don't think we study very well.
Thank you.
So are there, if you look at a postmenopausal versus premenopausal patient, is there a difference in circulating aldosterone? Does the absence of estrogen directly affect its production?
Yeah, so actually aldosterone levels in people do not increase with age, although mineralocorticoid receptor activity does. And so why is that? So again, I didn't talk very much about the whole section of my lab that studies smooth muscle cell mineralocorticoid receptors, but turns out smooth muscle cell mineralocorticoid receptors drive vascular aging. And what we recently discovered is that the expression of the receptor itself rises with age. And it's actually driven by inflammatory and oxidative stress transcription factors. And we all know that inflammation and oxidative stress rise with age. So there's actually more mineralocorticoid receptors in the vasculature in the older condition. And there's also some data from Eric Belenda-Chetimel's group in Georgia that endothelial levels of the mineralocorticoid receptor increase with obesity. So it may not only be the hormone, but it might actually be the receptor level. And so even in the absence of a rising aldosterone, there's an increase in activity with age and potentially with obesity that is driving the phenotype.
Right. Okay, we have one more question from Dr. McLaughlin.
Marvelous talk and really exciting work. I've studied the molecule estrogen for a long time, and I've looked at a couple steroids, but a lot of phytoestrogens and environmental estrogens and diethylstilbestrol. But your work brings up a question that I've had all this time and it is sort of a bedside to bench kind of approach. Why in the world do vertebrates make 3 ovarian estrogens that are different in their potency and their local action? And then why does the liver create catechol forms, which were more oxidatively reactive? 40% of circulating estrogen in women are dihydroxy derivatives on the A ring. Some are estrogenic, some are not. So why do you think, so many molecules of different potency? And the bedside question, which I tried to Google while you were talking, but I couldn't find it, is do the levels of estradiol and estrone and estriol change over time? And are one of those forms more prominent in menopause than not? I just don't know that. I don't know if anyone has. It's a naive question, but your work really sort of seeing how important it was and how sophisticated. And in endothelial, you're looking at one cell, so it's not even a structure like a uterus or a gland.
Yeah, so that's a great question. And I'll say, I'm not an estrogen expert. I came at this as an aldosterone expert. But I think it's a fantastic question. And I think it's a really important question for anything, is why do these things exist, right? There must be a reason that there are multiple different estrogen forms that are made, and they do have different potencies and act in different ways on the different receptors. And so they must be able to mediate different outcomes, and therefore you can then regulate them in different ways to mediate different outcomes. But I think, And then you get to your question about, so, what happens with menopause? And we know estrogen levels go down. I can't specify exactly, which type goes down more. But I do think that the underlying reason for that likely has something to do with what they do in younger people, right? Because as I talked about earlier, we weren't intended to live to be 90. And so, early people didn't really spend that much time postmenopausal, right? So I'm not sure that there's a specific role for those postmenopause, but I do think that there must be a reason why they were different premenopause, and they likely play a role in female physiologic function. And there's a lot more to female physiologic function than just pregnancy. But I can't specifically answer the different ones and why they're like that, but it's a great question.
Sarah, can I give my interpretation to John's question?
Of course.
Please.
I think I don't think it's known. The estrogen receptor is the oldest evolutionary steroid receptor. It appeared millions of years before any other steroid receptor. So it is the ancestor. So it's very important for a lot of functions. All these estrogens and derivatives are SERMs. They are selective estrogen receptor modulators. Each one of them induces a specific conformation of the receptor that will, in the nucleus, promote a different interaction with cofactors, and therefore will have a different transcriptional output. So I think that's the reason why we have a lot of them. Estrogen receptor is important, and you need lots of ligands that will produce a different receptor output.
But may I follow up? Then the frustrating part is every time you see a seminar, or if I give a seminar and I say I'm studying estrogen, I invariably mean estradiol 17 beta. And it would be very interesting, especially with Dr. Jaffe's system, to just probe it with 2 other estrogens. What would happen if instead of estradiol, you used estrone or estriol? From what you're saying, it would change the configuration and the activation of the receptor, and it might change how it interacts with the mineralocorticoid receptor, or what kinds of proteins you can pull down with that. So it just, I've never seen it done. I've never done it, but this seems like a really superb system to try to pull that and probe that.
Great point.
All right, Partha, did you have a question? I see your name there.
Yes, ma'am. My question is do you believe that this whole science also will be equally applicable for cerebral endothelium?
Oh, yeah. So that's a great question. So the cerebral vasculature, well, first I'll just take a big step back and say different vascular beds function differently. Even systemic vascular beds like the coronary versus the skeletal muscle vascular bed, for example. But the cerebral vasculature, I would say, is most distinct, and it functions quite differently. And we've done very little to look at that, although there are other groups like Andurance in Michigan who's looked at the role of the mineralocorticoid receptor in cerebral function, and it surely plays a role, but it's not exactly the same. And very recently, we did just start looking at the role of the mineralocorticoid receptor in the cerebral vasculature in terms of its regulation of vascular stiffness. And what we found is that changes that we see in the systemic vasculature are not necessarily seen in the cerebral vasculature. So the answer is, I suspect there's a role, but it's not the same.
Thank you.
Great question.
All right, thank you. And we'll take a 5-minute break, but all the trainees are invited to stay on the Zoom link and have a chance to ask more questions to Dr. Jaffe. Obviously, for our master's students, should be great to ask about clinical training as well. So thank you again for your talk, and have a good time with the students.
Thanks, everyone.